Okay, how are you today? I we not we but I definitely had a little accident in the last lecture on Monday, and that the lecture recording for some reason I don't know, I don't understand. It's all completely not recorded at all. That lecture was so. What am I going to do? Should we do it again, all over again? Sometimes actually that happens. So I don't know, or how can I just make it up for, or maybe just for that particular lecture, no recording is available. This another possible option. And okay, so regarding that, also, I have put up the new chapter on the snowboard today. And this particular chapter is not going to be covered in the classroom, mainly, actually. So this is all about chemistry. But of course, this one, the best thing about this particular chapter is ignore it completely. Because the main theme about this chapter is about the biological macromolecules. Like, so to be able to understand all four different types of macromolecules, including carbohydrate, lipids, proteins, and nucleic acid. So in order to do so, first off, we have to understand different types of chemical bonds. Okay. So we have to learn the covalent bond, hydrogen bond, and some other miscellaneous bond, the weak bonds like van der Waals, and hydrophobic interactions like blah, blah, blah. So all these things definitely, absolutely required for anybody who is seriously want to learn about, jump into the life science biology. Without knowing it, you cannot go further, because just right after this, we are planning on to jump into the tour of cells. So if you want to understand all different cells function with respect to different organelles and all different molecules functions, then you have to at least be able to understand and be able to distinguish the difference between proteins and lipids and carbohydrate, etc. Yeah. But on the other hand, I'm not going to heavily test this material on your upcoming exam, because that's not fair. If I were to test on you about the chemistry, because we have various different backgrounds, so that shouldn't be really fair game to evaluate you. I know, at least as far as I know, all of you are so very eager, enthusiastic. Even if you don't have any previous background, you want to learn. But this particular type of chemistry is going to be a little bit difficult for you to catch up within very short period of time. So that's why I thought about it. Okay, I'm going to put this material for only that Bogang, that makeup lecture, which is to be provided in the previous lecture recordings that I have already made in the previous semester. Okay, that is not very short material, but still, I'm not really going to. I only pick several, only very couple of, or two or three important points that out of this that I may test. Okay, which, and I'm going to let you know. I'm going to pick this, and probably I'd like you to know at least basic concept of this, and it will be on the upcoming midterm exam. Okay, that's what I'm going to do. So, having said that, so in the classroom, it's a person-to-person lecture, interacting lecture. After finishing this boring evolution chapter, we are directly going to jump into the cell part later. Okay, hopefully I should be able to. I hope be able to finish this chapter today, but I doubt it, because the way I move on. So anyway, let's give it a try. So no, this is not going to be. Yeah. So yeah, the day before yesterday we ended like after just briefly introducing these two terminology, micro versus macro evolution. These are nothing but like if you examine this figure, perhaps you should be able to understand the difference between macro. Macro means big, micro means a very mini scale, small scale evolution. Both of them are considered as an evolution, but there is a really big difference. Can anyone tell the difference by just looking at this figure under the two title headings of macro versus micro evolution? So if we try to connect this concept of evolution, whichever, micro or macro evolution, which of the two would be directly related with so-called speciation? Because evolution should end with, eventually should end, or sorry, I should say, culminate in the form of a speciation. Okay, that's the end of, that's the accomplishment of any single event of evolution. So in that case, which of the two, macro or micro, will generate speciation? After which? Yes, macro. Looks like macroevolution is the one generate the speciation. But then what about the microevolution? Here we are witnessing, we are actually observing the result of microevolution, because we are seeing all different individual as a result of a different traits. Some time ago, someone asked about what's the difference between the trait and allele. What's the allele? Allele is allele trait? Yes, it is. Allele, but allele is a little bit of a professional term, so many of you have not heard of this allele. All those majors should know. But allele is practically the synonymous to so alleles are different kinds of different version of any single gene is an allele. So is a trait, a definition of a trait. Under one such particular character, okay, you can have many different versions of traits. So that's why I can call both allele and traits are the same thing. The point is here, you are simply looking at different alleles or different traits, just like you are showing off, demonstrating it as an individual of a human species. You are demonstrating different type of alleles or traits. That's why you look all different. But eventually this type of thing may, if you are patient enough to give them such a long time, then this type of little mini variation event eventually may end up actual this macro evolution, then split into two distinct. Now you and I are totally different categories. No longer we used to belong within a same group. Okay, so this is at this stage, all these different individuals still belong to a same category, which we call a species. However, that as a macro scale, big scale evolution, now what you see as an end product is all different species of dinosaur. Okay, so that's the difference. Nothing too complicated. Okay, so So actually, I do have a very close friend or colleague or professor. I have to confess, even he is in this department. He's a diehard, I shouldn't say diehard, really faithful Christian as a biologist. He doesn't believe this, really. He doesn't believe at all about this macroevolution, but only kind of acknowledge, yeah, microevolution. Yeah, I respect that. That's his decision, that's his choice. Okay. But for me, it's very, also, hey, I am also kind of a pseudo Catholic. I'm a very bad Catholic. I never go to church, but still I'm a Catholic. I pray to God, but still, for me, it's very, very hard to deny this evolution because of my background. But anyway, so this microevolution by definition is this. The important thing is timeline. So this microevolution always you can observe. Okay. You can observe in your lifetime. Within the span of your lifetime, you can confirm something is happening. Because it can be quickly done. On the other hand, in other words, this macroevolution is speciation. And so nobody can witness something, this scale of macroevolution happening within your lifetime. You have to give at least something like tens of thousand years of long time. Nobody can live that long. You can only live up to 120, right? At most. So not possible. So maybe that is the reason why you cannot also you cannot be more familiar with this concept of macroevolution because you have never seen it. Yeah. Although in a scale of small creatures like viruses, you can, or even in some cases microorganisms like a bacteria, yes, you can. If you are lucky, you can witness something like similar to this macroevolution can even occur. You can check. But that's very rare extreme cases. So we cannot generalize this thing, little very rare event, into one example of such a macroevolution. So let's just put it this way. Even Charles Darwin somehow personally somehow thought about it and speculated some process of through this macroevolution, how modern day elephant emerged from the ancestral form of those ancestral elephants. Okay, there was kind of historic background. So this speciation, speaking of this speciation again, because those macroevolution will result in a new species, then back to original fundamental questions. Why can't those two different species biologically, for example, that those two individuals distinguished as two members of two different group of species, be able to interbreed? What is the reason? Why can't they? There are many reasons. So all of them, here I summarize briefly in this handout, lecture handout, that several occasions, different occasions of which this isolation can happen. So that is the reason that prevent these two individuals from two different species from mating. Okay. So later, if you have a little more time, you just may want to go over. The important thing is to simply try to emphasize the underlying, the big principle of this, the reason why the members of two different species cannot interbreed. Simply this. reproductive isolation. Then you sit down, what are different ways through which this so-called reproductive isolation can occur? You can think many different ways. Physically, but biochemically, physically, these two individuals cannot. It is impossible for them to have any such an activity. It's one such a reason. Or another very easy way you can easily imagine as an example of a reproductive isolation would be they are geographically too far away, isolated, so they never had any chance to meet. Okay, right? Just by simple texting doesn't cut it. Okay, so many excuse me, so many those reasons. Once you understand what is the whole underlying the big principle, which is that reproductive isolation, then you just go over, then everyone should be able to nod. Okay, that makes sense. Okay, that's it. I'm not going to go over to spend too much time in elaborating each of those little examples. Okay, nor would I put each details example on your exam. That's meaningless. Okay, so several examples. So pre-zygotic means before they fertilize. Post-zygotic means, you just think about it. Zygote. Zygote is a fertilized egg. Zygote. It's a big word, but in reality, it's just a simple sperm and egg. When they combine together, then it becomes zygote, fertilized egg. Okay. So pre-zygote versus a post-zygote, then you should be able to understand. Okay. Before they can do this event of fusion, sperm and egg, if there are some kind of obstacles, barriers exist, then we label them as a pre-zygotic barrier. You can think many different ways. Or post-zygote means even if they succeeded in this combining, but afterward, so you think, oh, we are all done, we made it. Not so fast, my friend. You will have to go through some other obstacles that you have to overcome. Then you failed. Then that we define as a post-zygotic barrier, reproductive barrier. That's it. So pre-zygotic habitat isolation, you know what that is. Okay. They live far away. So they have a seldom chance to get to see each other. Temporal. What do you mean temporal? Time-wise. So they appear, two species, two individuals living together in the same habitat, but still that doesn't mean if one always wake up in the midnight and then stay up until sunrise, and the other one just wake up normal, regularly, then there is no chance for these two individuals to see each other. That type of isolation, time-wise isolation, we can call temporal isolation, behavioral isolation. If these two individuals behave so oddly to each other, they don't have any feeling to have any intimate emotion to begin with, would there will be. So the behavior is also very important. There are so many examples of such behavioral isolation. Mechanical isolation is totally just physically unfit. They can't. For example, even dogs are, all different breeds of dogs at this point are considered as members of a same species. However, if you really think about it, Chihuahua, small, tiny Chihuahua, and big Great Dane, do you really think these two individuals can somehow make it? No way. Yeah. So those kind of physical barrier can also be there to separate them. And gametic isolation also, these two gamete, sperm and egg, so biochemically not compatible. So this fertilization event cannot take place because of this chemical reason. Then, so instead of trying to ask and try to have a really trivial question on each different thing, which, okay, is this a prezygotic barrier or what type of question? If you saw on your midterm exam, then it's not your fault. All you gotta do is blame the one who gave this type of a stupid question for you. That is not something that the point that you should carry with you to demonstrate that you understood this concept. Okay? So it's kind of briefly, although explained, so you should be able to understand with little difficulty. So I will just go zip through each detailed individual. So on the other hand, if you are particularly interested in any particular subtopic regarding this, then you are more than welcome to find out more in depth by searching. That's your also option or choice. And the post-zygotic barrier, these three are major examples of such a post-zygotic barriers. Then if you try to think of it, yeah, reduced hybrid viability, probably you should be able to understand. Okay. So you're able to have this mating, but the offspring, the hybrid, as a result of the mating, is somehow not doing so well. Very good example. Or reduced hybrid fertility also can sometimes it can be put together in the same category of this reduced viability or fertility, both, or in sometimes the separate reason. Top is viability is totally the health status, but the second example, reduced fertility, is they're doing very good physically, but for some reason no longer this hybrid is able to produce their own offspring, and then that should be the end of this event. The best known example of this reduced hybrid fertility is what? Can you guess? We even mentioned this particular example even in this class before. Yeah, this. The fertility means horse and donkey, they can mate, no problem. So mule is the consequence, but the mule cannot bear any children anymore. Mule is sterile. So what's the point? That's it. And actually the reason why these two, horse and mule, donkey, can somehow mate, but the hybrid, the mule, can no longer do this thing is because of their incompatible number of chromosomes. So when these two donate their half of the chromosome set, all of a sudden this mule have the odd numbered, incomplete pairs of a chromosome set. So this mule cannot generate its own gamete, sperm 1X. Because anytime those animals or plants who are engaged in sexual reproduction should make the gamete which have a reduced number of chromosomes in half. So like, think about this. As a human, we all these body cells of human contain how many chromosomes? 46. I mentioned this thing before. So how come you end up having 46 chromosomes? Because you got 23 from your mom and 23 from your father, right? So when you're up to make your own gamete, okay, then you reduce this chromosome number into half. So you have to, that's a must. That's your obligation. If not, that should be a big disaster if you don't do it. Okay. So you have to generate half the amount of chromosome in your sperm or egg. Okay. That's, do you still remember? That's the type of cell having half the amount of chromosome set? Haploid. You totally forgot about it? Oh, good for you. Good. So this haploid genome, you have 23, but then once you have this another gamete combined, then you will automatically restore this the double status, which we call the diploid amount of chromosome. But this mule, father and mother's chromosome sets numbers are different, so there's no way you can have an equal half amount of this gamete, because at least one chromosome should be broken into half totally physically if you really, really want to do that. That's why this mule, unfortunately, or maybe fortunately, become sterile. Okay. That is the one very famous, yes?
What difference of hybrid fertility is kind of hybrid breakdown?
A hybrid breakdown is different from this, the example of this rice. Hybrid, I do this again. Okay, so hybrid breakdown is very often observed in crops, plants. Okay? And here P stands for parental, two different type of breeds of rice. This F2 is a generation after the hybrid. So the direct consequence of this hybrid, this mating, should generate F1, but for some reason F1 is missing here, the photograph. But the thing is, F1 is really spectacular, impressive. It's tall, strong. Wow, hybrid. We call that, we recognize that as a hybrid vigor. Okay. So you're so excited about as a farmer. Wow, I got a new crop. And then you continue on the next generation, production of next generation, and then you end up this, always. There is some kind of mysterious barrier exists. So at the first generation, you have this hybrid, no problem, even much, much better hybrid than those two parent. So for the time being, temporarily, farmers get so excited about, so happy. So I got very big, sturdy hybrid. But then next generation, the mating of this F1 hybrid always generate really, really very poor or even sterile. So that's we recognize as a hybrid breakdown, because it is not exactly same as the more famous hybrid the fertility problem that we have witnessed in the case of donkey and the horse. So that's why it is treated a little bit differently. Okay? So this speciation, as we have already went through these different examples of isolation, reproductive isolation, the answer to this should be straightforward. What do you think? Speciation take place. This speciation can take place without? So like if there is a geographical separation, then it is so obvious that these two isolated populations, even though they were once regarded as a single species, but it will be just a matter of time that these two populations will evolve into totally two separate ones, then eventually becoming two new species. So geographical isolation is a very straightforward, easy to understandable mechanism by which two new species can arise, right? But the question is there any other example without involving this geographical isolation? Answer should be yes or no. Yes or no. Yes, of course. We have already seen several examples that doesn't really involve the geographical isolation. So it was just very dumb questions, but simply to check the degree of your understanding. Okay, how much you just got out of this. But the real reason is this terminology to go over. It is not really necessary, but there are some jargons, professional vocabulary regarding this speciation. One is called allopatric, the other one is a sympatric. Those who have this Western cultural language background, can you distinguish, can you explain the difference between allo versus a sim? No? Should I say S word then? Allo versus a sim. Yes, sim means the same. Allo means kind of a different. So Patrick means a geographical, the place. So this allopatric speciation means like a speciation occurring through a geographical isolation. In other words, we can usually just put as a other country. Allo means other. Those who took the biochemistry, allosteric control, allosteric inhibition, kind of allo means different, the other. Okay. So other country speciation, sim means the same, so same country speciation is a sympatric speciation. So it's just a matter of whether it takes the geographical separation or not is the difference, simply. Okay. Actually, this is the goal. The reason why I brought up these two, allo versus sim. Who cares about allo versus sympatric? But to bring up this very famous mechanism of generating this new species, even those members of the population living in the same location, is usually through this polyploidy. It's very similar kind of the occasion of the hybrid who is not fertile in the case of a horse and donkey mating. Like this chromosome number is not compatible. So similar thing can happen, like this polyploidy things. Because polyploidy, first off, polyploid, what's polyploid? The chromosome numbers is a very characteristic number to each species. So if I were to describe the characteristic is ploidy of human, then I would usually go with human 2n equals 46. That's the usual convention that we go by. Does everyone understand when I put this 2n equals 46? Yes? So probably chimpanzee, I guess I forgot, but maybe probably chimpanzees, this ploidy is 2n equals 48 type of. And so that's just big relief. No, I shouldn't say that's big relief, but there is a reason, the major reason why chimpanzee and human can not mate each other, because these chromosome numbers are totally different. So if you somehow try, there's no way you can generate any viable or fertile offspring. So this is ploidy. This is 2n. So if I instead write this is mathematically speaking, this is the same thing. 2n equals 46 and n equals 23. But in biology, describing ploidy, it is a different thing that we are talking about. N equals 23, we are talking about what? Haploid number. 2n equals 46, we are talking about diploid number. So probably, hopefully, you can understand when we are up to describe diploid versus haploid, the only thing you have to do is simply put n, using n as a basis. If you put number 2 in front of n, that means that indicates diploid. So what do you think the real definition of this n then? If you want to describe this thing, any individual cell in the setting of a diploid, then all you gotta do is a 2n equals 46 or 2n equals 4 or 2n equals whatever number. Then you always try to describe the diploid status. On the other hand, if you want to haploid status of any cell of any species organism, then you start with n equals 12, n equals 23, something like that. Because this n number is an intrinsic, unique number to each different species. So n number of Homo sapiens should be what? 23. So by having that particular n number, 2n equals 46 can be a human's diploid chromosome number, just like that. So here n, we can easily define n as a what? The number of haploid chromosome in each given species. This is the definition of n. It's not that difficult, or I don't have any. The only thing that you have to understand is whether whenever is something described in form of 2n equals something, n equals something, you should be able to distinguish, oh, this is something that indicating diploid cell, or this on the other end is something indicating haploid cell. As long as you are okay with, you should be all fine. Okay? Yes.
그러면 그 인간하고 침팬지가 서로 가까워서 그 염색체 수가 거의 딱 한두 개 바주체 이벤트 따져도 한두 개 차이 나니까 그러면 네안데르탈이 이 선사시대의 멸종은 네안데르탈인 염색체의 수도 추측할 수 있습니까?
Yeah, we all figured out that the chromosome number of Neanderthal is exactly the same as our humans. So 2n equals 46. Neanderthals too. Yeah. So at least as far as this chromosome number wise, there wasn't any barrier existed between our ancestor and Neanderthals. Okay? So this is how you end up accidentally on polyploidy cell. So So you start with your, so to speak, your body cells diploid number 2n. Okay. Whenever you try to produce gamete, you should reduce your chromosome number into half. So here, meiosis, the term, if you are not familiar about this meiosis, this is a special type of cell division occurring only during this gamete production. On the other hand, the, so to speak, the regular cell division, okay, we call mitosis. So this mitosis, through mitosis, you always generate from diploid cell, you generate always end up with same diploid cell. That's how you go through this mitosis. On the other hand, meiosis, in meiosis, you start from diploid cell, but your end result of this cell division is always a haploid cell. So if you carried out the meiosis, then the result should be haploid. So here, it should be N instead of 2N. That's the status of this gamete. Either it's an egg or sperm. But some kind of error happened. So they failed in reducing their chromosome number through this procedure. Okay, so you end up still 2N. Hey, what the heck? Life is just, why do you have to be so freak about being a haploid? Life is too short. Take it easy. What's wrong with this having more number of chromosomes? More the better, isn't it? Then you just bravely go through. No, in this case, no. Because let's say these two diploid gamete somehow were able to, luckily this one diploid gamete was lucky enough to meet another diploid gamete. Both of them should have been haploid, but whatever. And then the result of this fertilization should be 4N. That's what we are talking about, the polyploidy. If this polyploidy, here, what's the N number in this cell? Probably that's something that I have to. In this original cell, tell me, what is the N number of this cell? N is 4? No, here, this original 2N cell. What's the N number? Just like human cell, the N number is 23, isn't it? Human cell, 4N, 2N equals 46. So if someone asks it, what's the N number of, what's, oh, I'm sorry, my pronunciation problem. Okay, so this humans N number is 23, whereas what about this? What's the N number of this particular cell? Three? Yeah, that's correct. How did you figure it out? Yeah, you had a 6 chromosome. Oh, that's why you just simply automatically 6, 2N equals 6. Okay, that's exactly. So here, I would like you to understand this concept of N, 2N. Why this cell is regarded as 2N. Here, graphically speaking, this cell is being described as having 3 different kinds of chromosomes. 3 different kinds of chromosome, each of these 3 different kinds of chromosome having a partner. like color coded in single particular color. Okay, so this purple chromosome, the reason why this purple chromosome has a two chromosome of purple is because one came from father and the other one came from mother. So that's how exactly describing this 2N status. So if this cell became, instead of being 2N, became just one, just single N haploid cell, then how would you draw this cell? One cell having 3 chromosomes of 3 each different colors retaining. But none of them should have a double, like more than one color chromosome, same color chromosome. That should be the definition of haploid. Sometimes some of you may just laugh at me. Oh, that's so easy, so childish you are going over this. Sometimes a lot of you actually make mistake even on the real exercise to correctly describe the status of actual real haploid cell. That's why I am stressing one more time about this. So anyway, this 4N cell, since this cell's N number is 3, this 4N cell would have a little difficulty in generating correct gamete later on. Because this 4N cell will end up, the chances are, a lot of, no. My point is, this 4N cell now generate half the number of chromosomes, which will become 2N cells. Okay, but this has to find another same exact number of chromosomes. But then, usually the regular gamete will have this N haploid genome. So if these two somehow try to combine, then you have this 3N. And look at this. In the 3N status of, do you have every chromosome have its own partner? Like those partner chromosomes, by the way, do you know how they are called? The same chromosome, basically the partner chromosome. One came from father, the other one came from mother. What's the name we call those chromosomes? You forgot? Homologous chromosome. Okay. So, in other words, this purple and this purple, these two chromosomes are homologous chromosomes to each other. And there is one more homologous chromosome, like odd numbered chromosome. It shouldn't have been there, but that's the major reason why this cell will be sterile. So, animals, polyploidy is extremely rare, really, because of this problem. The only observed example of polyploidy is some rodent, like small animal, found in South America, having 4N instead of 2N. Usually animals are 2N diploid. Vast majority of animals are all diploid organisms. But here, 4N means tetraploid animal is extremely rare. But on the other hand, the plants is much more tolerant to this unusual chromosome number. And in some cases, we exploit their tolerance to this polyploidy to generate like better quality of the crop product. So polyploidy, because having more chromosome in the case of plants, get to produce bigger fruit and their bodies are larger. So those are very desirable traits, agricultural trait for farmers. So we would welcome those unusual status. So look at this. The vast majority of watermelons are tetraploid, usually. Or even some watermelons are hexaploid. Regular, I shouldn't say normal, but regular, standard, more standard, original, diploid watermelon would be much smaller, taste less desirable, less tasty, and blah, blah, blah. Look at this strawberry, compare this. Diploid strawberry and tetraploid strawberry. Which one would you rather pick on the supermarket? Obviously, okay, I'd rather. This has a much more attractive market value. Kiwis too. Here, tetraploid kiwi versus diploid kiwis, obviously big differences. Even you can also manipulate this, like on the market, although it is not really that popular, but there are some seedless watermelons available. The reason they are seedless is they are triploid. Triploid, now you understand, the odd numbered chromosome set will be sterile. Same goes with bananas. So those odd numbered chromosome set will generate this sterility. Then sometimes we love to have those sterile fruit products. Okay, so like I said, on the other hand, in animal, this is probably one such rare exception that living as a tetraploid animal. Other than that, most of those found turn out to be simple diploid.
What kind of polyploid animal?
This is one example. Only one example we could find polyploid. So tetraploid. 4N. Gradualism and punctuated equilibrium. The mode of evolution, speciation, tempo, gradual versus punctuated. This is two differences. This is probably a lot of you have thought about the mode of the way this new speciation emerged from the single common ancestor. But sometimes, so both of these two modes exist in nature. Some type of speciation occur so slow and gradually the difference, the difference gap become wider and wider as time goes. But some other occasions, so sudden, gradual, quick change, very big change occur. And then that change is somewhat consistently maintained throughout the long period of the time. That's we call the punctuated equilibrium, as opposed to gradual. We call it gradualism. Both of them are the real life examples. If you think, in what occasion this occur and on what occasion this will occur, is you can speculate. Perhaps, maybe, no, I wouldn't. I would search. At the moment, momentarily, I thought about having this type of asking the question on your exam. What's the what type of different occurrence event would have made these two differences, but never mind. So these two, at least let's just put it this way simply. Both of these two different modes exist. Okay? Human origin, I told you, I doubt that I would be able to finish this thing within. Or should I just simply skip the question and answer session today and then go through this human evolution today? Yes? Okay. Forget about the question and answer session. Okay, later, maybe I will just deal with that on next Monday. So today, I'm going to continue on just finishing this thing. So origin and evolution of humans, let's see. These are not really important. You can simply go through on your own about our relatives, cousins. That's not the point. Even they are not on your exams, so don't you worry about. So, okay, here's the point. The most famous, the way of human evolution, starting from so-called Australopithecus, and then gradually becoming now this Homo sapiens. You notice that in this is actually a lot of scientists distinguish these two, Neanderthals and Homo sapiens, as a member of the same species. So scientific name is Homo sapiens, both of them, Homo sapiens and Homo sapiens. Only the subspecies names are different. Like in the case of our own ancestor, and us too, Homo sapiens sapiens, whereas this all extinct Neanderthals are called Homo sapiens neanderthalensis, indicating that they were able to interbreed together, so they should be regarded as members of the same species. Before, in the past, actually, they were called Homo neanderthalensis, whereas we were called Homo sapiens, totally indicating these are the two separate species. But then it turns out that, hey, they mated and they produced children together. So like, how can we separate these two as two individual distinguishable species? We shouldn't. It's probably the reaction, outcome of this change. But anyway, so these are not that important. The thing is, the actual origin turns out to be this out of Africa. There are a couple of other theories trying to explain the origin, how all those different humans living today on Earth, where they came originally from. But like after thorough investigation and confirmation, it turns out that this particular theory called out of Africa theory is the one actually correct theory, which explains that all the existing today's humans are all derived originally from Africa. So before that, outside of Africa, there were no human living. So all those humans were originally from the Africa. Then differentiated. So those small group of migrants came from Africa, moved out to all different places on Earth, and have differentiated to establish their own unique ethnic group, like races, is what the current theory explains. So having said that, then what do you think? Today, if you examine genetic diversity of everywhere on Earth, let's say between two groups inside of within Africa and outside of Africa, which area do you think that genetic diversity is higher? higher genetic diversity exists outside of Africa if you examine all those human DNA sequences to determine how different they are, and then compare those diversity with those you examine within Africa. Those who you're assuming that those still living in Africa originally mostly, more or less, are the one who stayed there. Okay, although a lot of them actually temporarily moved to some other location like America, and then when came back, some of them, yeah, that's true. But anyway, overall, what do you think? Africa or outside of Africa? No. Inside of Africa has more genetic diversity. Why?
인류가 더 유래했을
Why? I mean, yeah, we originate from there, but does that mean really the genetic diversity should be there more? I mean, less genetic diversity found outside of Africa actually indicate one thing. Assuming the history of humans, the time span, their lifespan stayed the same because they were all originated from a single human, then what do you think? Because that means only very small number of, relatively speaking, small number of people moved out of Africa. So there in the mainland, the African mainland, still there are vast majority of the people, human, remain and stayed there. So to begin with, the genetic diversity to begin with was much higher than, because the smaller the people, you would have much less genetic diversity to start with. That's exact kind of a proof that, originally the number of people who moved out of Africa was not really such a huge number of big gang of people, but only very small, relatively speaking, small number of people moved out of it and then became the ancestral humans of all the humans living outside of this, currently outside Africa. So anyway, whatever those called the Homo erectus, some very small population of Homo erectus migrated out of. Actually, that migration event took place twice. Earlier event of migration established that Neanderthals, basically, on Europe, and certain part of the Asia. And then much later, once again, much later, about this 150 or 200,000 years ago, another small group of humans migrated out of Africa. I cannot stand this shitty place anymore. I will just move out of this. And then they did it again now. And then they obviously were encountered with the existing inhabitants on Europe already. They are Neanderthals. In other words, these two new groups coexisted actually for quite some times over the area like Europe and Eurasia, basically. Okay, and that's when this interbreeding between our ancestor Homo sapiens and Neanderthals happened. So this is what something that diagram indicating this out of Africa theory. Nothing special about it. So this coexistence is the point, and during which this events of mixed breeding happened. And this is projected, and the migratory route they took. It turned out that, and yes.
Why Neanderthals and Denisovans are extinct?
Extinct?
Why Neanderthals?
The question is, yeah, good question. The very good question is probably one couple of $1 million questions, but still people don't know why are we the only one survived out of all different recent Homo sapiens. Not only that, Neanderthals, Denisovans, Heidelbergensis, fluorescens, all different kind of modern day humans. Evidence of their existence were found. And also another thing is all these people showed the evidence of interbreeding among them. So it's not only the Neanderthals and us, but also all these exchange their genes to some degree. But only we are the one. Maybe we killed all of them? We don't know. Well, we are the one who just fit the best? We don't know. Looks like Neanderthals from the evidence of the fossils and other clues. Neanderthals should be able to do very well also, as good as our ancestors in many aspects, but still we don't know. So since we do have so many different lineages, so how do we identify which one's which came first and which one is our closest relatives or any kind of such thing? How do we determine those? Simply by comparing the shape of bones and the imaginary body shape is not enough to give a very correct, accurate estimate. For that, this DNA sequence comparison is the best, because later I will go over once again why theoretically this DNA sequence comparison can be used as a really, really accurate measure for this relative relatedness, okay, among different, not only humans, but for many, many different organisms. DNA sequence comparison is really, really the most reliable indicator by which we can determine. Probably we don't have enough time today to go over that. Maybe I should finish that one by tomorrow, by next Monday, but I will go over definitely. But anyway, among those sequence comparison, especially this mitochondrial DNA and human Y chromosome DNA sequence have been very useful in determining this relative close species or individuals among very similar, closely related individuals like humans. So, for example, if you want to indicate, okay, we are Koreans, some of you are not, but we are Korean as a Korean ethnicity. Which other ethnicity is genetically the most closely related with us? How do we determine those? Of course, you can read the entire 3 billion alphabet of DNA and then determine, but that would be really a waste of time and money for such event. So then quickly and more accurately you can compare the mitochondrial DNA sequence or Y chromosome. Mitochondrial DNA? Did mitochondria have DNA? Yes, they do. Mitochondria have their own DNA. So these type of occasions, this mitochondria and Y chromosome DNA are particularly useful in such relationship determination. Why? The reason, of course, there is a reason. Mitochondrial DNA is special in that, first off, mitochondria as an organelle contains, surprisingly, if you didn't know, surprisingly contain their own DNA. Okay, So it's like in our cell, eukaryotic cell, the nucleus is not the only place where you can find the DNA. Mitochondria and also chloroplast, although we don't have any chloroplast, but chloroplast too have their own DNA. So this mitochondrial DNA having several multiple copies within, this is a kind of a cartoonic version of the mitochondrial cutoff diagram. So all these single tiny circles represent their own DNA. Their DNA is, for some reason, very peculiar in that donut-shaped circular DNA, unlike our human DNA or any other eukaryote DNA, which is linear DNA. So this DNA, the usefulness of mitochondrial DNA is it has no recombination. What's recombination? Probably you would ask, of course. The DNA in your nucleus as a eukaryote undergo this very interesting exchange thing. Whenever they try to produce gamete through meiosis, meiosis is the only event that these two chromosomes, one is blue and the other one is red, these are actually the homologous chromosome set. The blue must be coming from father's origin and red is coming from mother's. If you don't like a red one, yeah, I can switch. Okay, so red is from a father, whatever. Okay, so when they align just right before their actual cell division, they this kind of really odd things like a crossing and then exchange this, so it ends up swapping some part of this. This is called what we call as a recombination. This recombination shouldn't generate any dangerous problems, because these two chromosomes are homologous anyhow. They are same kind of chromosome, so just one part of this thing is like a replaceable from the other homologous part, the same region. And actually this way, it is one way of increasing the genetic variation by this, like shuffling. Whenever you play poker or go stop, you always shuffle your cards, right? That's, who are you? So that's one way. Is that this recombination is not desirable for correctly estimating the relative, this relatedness between two individuals. Perhaps I should provide a further, more detailed, more kind explanation about this maybe next Monday, because now time hits 4:13 now. So I will shut up now, and then just simply I will wish you have nice weekend, and then see you on Monday, and then continue on this. Bye.
왜 인류와 공존하고 교배까지 했다는 그런 네안데르탈인, 데니소바인, 화이델베르크인, 플로레스인은 왜 다 멸종?